All right, so today we got mitosis and meiosis by me. Okay, so the purpose of each the goal of mitosis is one parent cell will get divided into two identical daughter cells to keep it consistent. And then the purpose of meiosis is to create four daughter cells total. And the purpose is to have half the chromosomes and a quarter of the DNA in all of it, and to increase variation, genetic variation, and all that. But then specifically related to mitosis, they use somatic cells, which are non-sex cells, which have all 46 chromosomes in it, while meiosis includes gametes, which only have the 23 chromosomes, which are sex cells, which include sperm and eggs. But when it comes to chromosomes, each chromosome is like a different gene locus where the genes are in a specific location on the chromosome. So it varies between people, such as Dr. Phil, Bailey, whatever that is, Mr. Hellfire. They all have different gene locuses for different their chromosomes, which makes them not look all the same, but they're all different. Now I'll talk about more specifically mitosis, how we divide into two identical daughter cells. And there's four phases of it, but before, which includes PMAT, so prophase, metaphase, anaphase, telophase. And before that, it's the process of interphase, which includes specifically more G2, where the DNA has been replicated and the chromosomes are ready to be made for prophase. So for prophase, the chromosomes they're crossed over into two sister chromatids, which are two identical chromatids, which are connected in the middle by a centromere, which is just a little gap. And later on, they're connected with the kinetochores, which is the spindle fibers. It's still in this stage, the spindle fibers they're starting to form, and the poles as well, the centromeres, which are the poles. And in this stage, the nucleoli are starting to disappear. The chromatin they're becoming that chromosome again. They're becoming that X, where the centromeres in the middle, or the mitotic spindles become to form like the little lines that go through the centromeres from both pole to pole. And then the centrosomes move apart as well. In this stage, metaphase, the centrosomes are fully set up. The spindle fibers have been totally formed, and the chromosomes are lined up. So the spindle fibers go directly through the centrosome from pole to pole, and then the nuclear membrane starts to fade, especially in this phase. It does start to fade in prophase, but it starts to fade even more in this. When it goes to the center, it's right next to the metaphase plate, which is where they line up. It's not like a real thing, but it's where they line up next to each other. And then the sister chromatids, they face either side. So if the left part of the X faces the part facing west, and the right chromosome faces the part facing east. And then in anaphase, the chromosomes, like the sister chromatids, pull apart, and they pull towards the different poles of the centrosomes, and the nuclear membrane is totally gone at this point. And the poles are moving apart. So it elongates the cell, and it's like stretching it, so it's more of like an oval instead of a circle. And then those kinetochores where the hook's kind of shortening because of since the spindle fibers are shortening. And then in telophase, the cell is starting to divide into to become two daughter cells. And then those two nuclei begin to form, and then so do the nuclear envelopes. And then the nucleoli appear as well. And then the X chromosomes that then became like the looking arches looking thing started to become chromatin again. So they were just straight lines. And they were like all uncoiled. And then after this would be cytokinesis, which is the fully division of the two cells. Now, specifically more meiosis is where the gametes, where the 23 chromosomes come into play. And before, technically, or why doesn't it work? When it comes to prophase, right? Before this, the chromosomes, they're not crossed over yet, which is you can see the top with the two golf balls are switch colors. So the red one's on the white chromosome now, and then the white one's on the red one. This is the process of crossing over, where basically like the top two little bits are switching sides just to create more variation among the sex cells. But technically, before prophase, it's not actually crossed over. But in this before prophase, there's a process called synapsis, which basically brings those two chromosomes together. And then where the X's are touching is the chiasmata, which is where they just kind of touch throughout the entire process until they get split up in anaphase. And then basically once those are crossed over, they're known as tetrads, since there's technically four sister chromatids there. Not four sister chromatids. There's two sister chromatids per chromosome, so 2 times 2 is 4. And the poles are starting to form, that nuclear envelope is starting to go away as well. In metaphase, the spindle fibers are there again, the centrosomes are there to pull the chromosomes towards it. And they line up again into this phase where the nuclear envelope is still there, but eventually it'll fade away. But this is only the first half of meiosis. And then in anaphase, the same thing. It becomes like that arch thing. The sister chromatids begin to split apart and get pulled towards one of the centromeres. Then, after this, which is telophase, the two new cells begin to form, creating where the poles are still there, but they're kind of irrelevant at this point. Where after this, there's no really cytokinesis. There's cytokinesis again, where the two cells are fully divided, but this isn't over yet. So now we're in prophase 2, which the homologous chromosomes are in different thing. They're in two different cells now. Yeah, they're all four of them are variated, since they don't all have the same gene at the top. In metaphase now, there's more it's basically a repeat of mitosis, where the poles are formed, the spindle fibers are there going through the center of the centromeres. Then it's pulling apart the chromosomes, which happens in anaphase, where the arch becomes a thing again, just pulling it apart, letting the spindle fibers become shorter. And then in telophase, essentially, there's 4 cells that are going to form, where it unwinds again to become more chromatin. And so it's just more strands, and all of those cells are variated differently. So the whole point of the goal of meiosis has been complete, since it's half the chromosomes per thing and a quarter of the DNA per daughter cell from the parent cell, because there's 4 of them. And after this, the cytokinesis divides it too. So it's 4 daughter cells that are all variated. But more specifically about metaphase here, there's 2 laws that Mr. Mandel came up with. And the one's called the law of segregation, and one's called the independent assortment. So an independent assortment, the homologous chromosomes, it doesn't matter what side it's on. Like when it comes to line up, it doesn't really make a difference. So it could be one could be on one side, one could be on the other. And it doesn't have to be 2. There could be 20, there could be 40. But it doesn't matter what side it's on. It's not going to change the outcome of the variation. It's still going to be varied no matter what. And then more specifically in anaphase, there's the law of segregation. They said that the alleles segregate from each other. It's only one allele is present in each gamete, which you can see here. Since there's only one here, these are all different from each other. None of these are the exact same. This one has one red ball. This one only has white. This is all white. This is all red. So it ensures that they're all different at the end. And that's all I got. Appreciate the time.